Ooh, that's a bit far. I'll move you closer. Sure. Good shiced. Okay. Okay. Next chapter, y'all. Curiouser and Curiouser. That sounds so wrong. Curious. Curiouser and Curiouser. Alright. Okay. So let's go forward. We've got one quote. You're under a lot of stress, my dear. You haven't really got anything wrong with yourself. We'll give you an antidepressant. And that is Barry Marshall on the treatment of women with gastritis, a precancerous lesion in the 1960s. Whew, let's go. The classification of tobacco smoke as a potent carcinogen and the slow avalanche of forces unleashed to regulate cigarettes in the 1980s is rightfully counted as one of cancer prevention's seminal victories, but it equally highlighted an important lacuna in cancer epidemiology. Statistical methods to identify risk factors for cancer are, by their very nature, descriptive rather than mechanistic. They describe correlations, not causes, and they rely on a certain degree of foreknowledge. To run a classical case control trial to identify an unknown risk factor, paradoxically, an epidemiologist must know the questions to ask. Even Dahl and Hill, in devising their classic case control and prospective studies, had relied on decades of prior knowledge, centuries of one counts John Hill's pamphlet about the possible link between tobacco and cancer. This does not diminish the incredible power of the case control method. In the early 1970s, for instance, a series of studies definitively identified the risk factor for a rare and fatal form of lung cancer called mesothelioma. When mesothelioma cases were compared to controls, this cancer appeared to cluster densely in certain professions: insulation installers, firefighters, shipyard workers, heating equipment handlers, and chrysotile miners. As with pot and scrotal cancer, the statistical confluence of a rare profession and a rare tumor swiftly identified the causal agent in this cancer: exposure to asbestos. Tort litigation and federal oversight soon followed, precipitating a reduction in the occupational exposure to asbestos that, in turn, reduced the risk of mesothelioma. In 1971, yet another study identified an even more unusual carcinogen, a synthetic hormonal medicine called diethylstilbestrol, or DES. DES was widely prescribed to pregnant women in the 1950s to prevent premature deliveries, although it was only of questionable benefit in this regard. A generation later, when women with vaginal and uterine cancer were questioned about their exposures to estrogens, a peculiar pattern emerged. The women had not been exposed to the chemical directly, but their mothers had been. The carcinogen had skipped a generation. It had caused cancers not in the DES-treated women, but in their daughters exposed to the drug in utero. But whether the behavior or exposure responsible for the cancer is completely unknown. What if one did not even know enough about the natural history of mesothelioma, or the link between estrogen and vaginal cancer, to ask those afflicted about their occupational history, or their exposure to asbestos and estrogen? Could carcinogens be discovered a priori, not by the statistical analysis of cancer-afflicted populations, but by virtue of some intrinsic property of all carcinogens? Bad start. Well, that's our first break. But we must keep on going. I'm gonna put the book here. In the late 1960s, a bacteriologist geneticist named Bruce Ames at Berkeley working on an unrelated problem, stumbled on a test for chemical carcinogens. Ooh. Some clothes just fell down, sorry. Oh yeah. Fuck, I missed. Fucking dusty bitches. Alright. Ames was studying mutations in Salmonella, a bacterial genus. Salmonella, like any bacteria, possesses genes that allow it to grow under certain conditions. A gene to digest galactose, for instance, is essential for a bacterium to survive on a petri dish when the only sugar source is galactose. Ames observed that the mutations in these essential genes could enable or disable the growth of bacteria in a petri dish. A strain of Salmonella normally unable to grow on galactose, say, could acquire a gene mutation that enabled this growth. Once growth enabled, a single bacterium would form a minuscule colony on a petri dish. By counting the number of growth-enabled colonies formed, Ames could quantify the mutation rate in any experiment. Bacteria exposed to a certain substance might produce 6 such colonies, while bacteria exposed to another substance might produce 60. This second substance, in other words, had a 10-fold capacity to initiate changes in genes, or a 10-fold rate of mutation. Ames could now test thousands of chemicals to create a catalog of chemicals that increase the mutation rate. Mutagens, and as he populated his catalog, he made a seminal observation. Chemicals that scored as mutagens in his test tended to be carcinogens as well. Dye derivatives, known to be potent human carcinogens, scored floridly, causing hundreds of colonies of bacteria. So did X-rays, benzene compounds, and nitrosoguanidine derivatives, all known to cause cancers in rats and mice. In the tradition of all good tests, Ames' test transformed the unobservable and immeasurable into the observable and measurable. The invisible X-rays that had killed the radium crows in the 1920s could now be seen as revertent colonies on a petri dish. Ames' test was far from perfect. Not every known carcinogen scored in the test. For instance, asbestos sprinkled on the disabled Salmonella did not increase the number of mutant bacteria. In contrast, chemical constituents of tobacco smoke did cause mutation in the bacteria, as noted by several cigarette manufacturers who ran the test and, finding it disconcertingly positive, quickly buried the results. But despite its shortcomings, the Ames test provided an important link between a purely descriptive approach towards cancer prevention and a mechanistic approach. Carcinogens, Ames suggested, had a common distinctive functional property. They altered genes. Ames could not fathom the deeper reason behind this observation. Why was the capacity to cause mutations linked to the ability to induce cancer? But he had demonstrated that carcinogens could be found experimentally, not retrospectively, but by prospectively identifying chemicals that could cause mutations in a rather simple and elegant biological assay. And that's our second break, and I haven't eaten breakfast yet, so I am really quite hungry, but yeah. Chemicals, as it turned out, were not the only carcinogens, nor was Ames' test the only method to find such agents. In the late 1960s, Baruch Blumberg, a biologist working in Philadelphia, discovered that a chronic smoldering inflammation caused by human hepatitis virus could also cause cancer. A biochemistry student at Oxford in the 1950s, Blumberg had become interested in genetic anthropology, the study of genetic variations in human populations. Traditional biological anthropology in the 1950s mainly involved collecting, measuring, and categorizing human and anatomical specimens. Blumberg wanted to collect, measure, and categorize human genes, and he wanted to link genetic variations in humans to the susceptibility of diseases. The problem, as Blumberg soon discovered, was the lack of human genes to be measured or categorized. Bacterial genetics was still in its infancy in the 1950s. Even the structure of DNA and the nature of genes was still largely undiscovered, and human genes had not even been seen or analyzed. The only tangible hint of variations in human genetics came from an incidental observation. Proteins in the blood, called blood antigens, vary between individuals and were inherited in families, thus implying a genetic source for this variation. This blood proteins could be measured and compared across populations using relatively simple tests. Bloomberg began to scour far-flung places in the world for blood, drawing tubes of serum from Fulani tribesmen in Africa one month and Basque shepherds the next. In 1964, after a brief tenure at the NIH, he moved to the Institute for Cancer Research in Philadelphia, later renamed the Fox Chase Cancer Center, to systematically organize the variant blood antigens that he had cataloged, hoping to link them to human diseases. It was a curiously inverted approach, like scouring a dictionary for a word and then looking for a crossword puzzle into which that word might fit. One blood antigen that intrigued him was present in several Australian Aboriginal subjects and found frequently in Asian and African populations, but was typically absent in Europeans and Americans. Suspecting that this antigen was the fingerprint for an ancient genetic factor inherited in families, Bloomberg called it the Australia antigen, or AU for short. In 1966, Bloomberg's lab set out to characterize the Aboriginal antigen in greater detail. He soon noted an odd correlation. Individuals carrying the AU antigen often suffered from chronic hepatitis and inflammation of the liver. These inflamed livers, studied pathologically, showed signs of chronic cycles of injury and repair, death of cells in some pockets, and compensatory attempts to repair and regenerate liver cells in others, resulted in scars, shrunken, and burnt-out livers, a condition termed chronic cirrhosis. A link between an ancient antigen and cirrhosis suggested a genetic susceptibility for liver disease, a theory that might have sent Bloomberg off on a long and largely fruitless tangent. But a chance incident overturned that theory and radically changed the course of Bloomberg's studies. The lab had been following a young patient at a mental disability clinic in New Jersey. Initially, that man had tested negative for the AU antigen, but during one of his serial blood draws in the summer of 1966, his serum suddenly converted from AU negative to AU positive when his liver function was measured and acute fulminant hepatitis was discovered. But how could an intrinsic gene suddenly cause seroconversion and hepatitis? Genes, after all, do not typically flicker on and off at will. Bloomberg's beautiful theory about genetic variation had been slain by an ugly fact. AU, he realized, could not mark an inherent variation in the human gene. In fact, AU was soon found to be neither a human protein nor a blood antigen. AU was a piece of viral protein floating in the blood, the sign of an infection. The New Jersey man had been infected by this microbe and thus converted from AU negative to positive. Did Bloomberg, when he collected all that blood, did he spread that shit himself? Was he responsible for infecting all those Aboriginal people? Whatever. Bloomberg now raced to isolate the organism responsible for this infection. By the early 1970s, working with a team of collaborators, his lab had purified particles of a new virus, which he called hepatitis B virus, or HBV. The virus was structurally simple, roughly circular, about 42 nanometers in diameter, one of the smallest DNA viruses that infect humans. But the simple structure belied extraordinarily complex behavior. In humans, HBV infection caused a broad spectrum of diseases, ranging from asymptomatic infection to acute hepatitis and chronic chronic cirrhosis in the liver. The identification of a new human virus set off a storm of activity for epidemiologists. By 1969, Japanese researchers, and subsequently Bloomberg's group, had learned that the virus was transmitted from one individual to another through blood transfusions. By screening blood before transfusion, using the now familiar AU antigen as one of the early biomarkers in serum, the blood-borne infection could be blocked, thereby reducing the risk of hepatitis B. But another illness soon stood out as linked to HBV, a fatal insidious form of liver cancer endemic in parts of Asia and Africa that arose out of scarred, ashen livers often decades after chronic viral infection. When cases of hepatocellular cancer were compared to controls using classical statistical methods, chronic infection with HBV and the associated cycle of injury and repair in liver cells stood out as a clear risk factor, about 5 to 10-fold the risk for uninfected controls. HBV then was a carcinogen, although a live carcinogen, capable of being transmitted from one host to another. Man, okay. I just scratched something that hurt like hell. Okay. Alright. The discovery of HBV was an embarrassment to the NCI, the Institute's highly targeted and heavily funded special virus cancer program, having inoculated thousands of monkeys with human cancer extracts, had yet to find a single cancer-associated virus. Yet a genetic anthropologist exploring aboriginal antigens had found a highly prevalent virus associated with a highly prevalent human cancer. Bloomberg was acutely aware of the NCI's embarrassment and of the serendipity in his work. His departure from the NIH in 1964, although cordial, had been driven by precisely such conflicts. His interdisciplinary curiosity had chafed against the discipline-determined rigidity of the constituent institutes, among which the NCI, with its goal-directed cancer virus hunt, was the worst culprit. Worse still, for the strongest enthusiast of the cancer virus theory, it appears as if Bloomberg's virus itself was not the proximal cause of the cancer. The inflammation induced by the virus in liver cells and the associated cycle of death and repair appeared to be responsible for the cancer, a blow to the notion that viruses directly cause cancer. But Bloomberg had little time to mull over these conflicts, and he certainly had no theoretical axes to grind about viruses and cancer. A pragmatist, he directed his team towards finding a vaccine for HBV. By 1979, his group had devised one. Like the blood screening strategy, the vaccine did not, of course, alter the course of the cancer after its genesis, but it sharply reduced the susceptibility of HBV infection in uninfected men and women. Bloomberg had thus made a critical link from cause to prevention. He had identified a viral carcinogen, found a method to detect it before transmission, and thus found a means to thwart transmission. My reading skills are fucked. That was another break, by the way. Okay, but we must keep going. The strangest among the newly discovered preventable carcinogens, though, was not a virus or a chemical, but a cellular organism, a bacterium. In 1979, the year that Bloomberg's hepatitis B vaccine was beginning its trial in America, a junior resident in medicine named Barry Marshall and a gastroenterologist, Robin Warren, both at the Royal Perth Hospital in Australia, set out to investigate the cause of stomach inflammation, gastritis, a condition known to predispose patients to peptic ulcers and to stomach cancer. For centuries, gastritis had rather vaguely been attributed to stress and neuroses. In popular use, the term dyspeptic still refers to an irritable and fragile psychological state. By extension, then, cancer of the stomach was cancer unleashed by neurotic stress, in essence a modern variant of the theory of clogged melancholia proposed by Galen. But Warren had convinced himself that the true cause of gastritis was a yet unknown species of bacteria, an organism that, according to dogma, could not even exist in the inhospitable acidic lumen of the stomach. Since the early days of medical bacteriology, over 100 years ago, Warren wrote, it was taught that bacteria do not grow in the stomach. When I was a student, this was taken as so obvious as to barely write a mention. It was a known fact, like everyone knows that the earth is flat. But the flat earth theory of stomach inflammation made little sense to Warren. When he examined biopsies of men and women with gastritis and gastric ulcers, he found a hazy blue layer overlaying the crater-like depressions of the ulcers in his stomach. When he looked even harder at that bluish layer, he inevitably saw spiral organisms teeming within it. Or had he imagined it? Warren was convinced that these organisms represented a new species of bacterium that caused gastritis and peptic ulcers, but he could not isolate the bacteria in any form on a plate, dish, or culture. Others could not see the organism. Warren could not grow it. The whole theory, with its blue haze of alien organisms growing above craters in the stomach, smacked of science fiction. Barry Marshall, in contrast, had no pet theory to test or disprove. The son of a Calgary boilermaker and a nurse, he had trained in medicine in Perth and was an unwedded junior investigator looking for a project. Intrigued by Warren's data, although skeptical of the link with an unknown phantasmic bacteria, he started to collect brushings from patients with ulcers and spread out the material on petri dishes, hoping to grow a bacterium. But as with Warren, no bacteria grew out. Week after week, Marshall's dishes piled up in the incubator and were discarded in large stacks after a few days of examination. But then serendipity intervened. Over an unexpectedly busy Easter weekend in 1982, with the hospital overflowing with medical admissions, Marshall forgot to examine his plates and left them on the incubator. When he remembered and returned to examine them, he found tiny translucent pearls of bacterial colonies growing on the alar. The long incubation period had been critical. Under the microscope, the bacterium growing on the plate was a minuscule, slow-growing, fragile organism with a helical tail, a species that had never been described by microbiologist. Okay. Warren and Marshall called it Heliobacter pylori. Heliobacter for its appearance, and pylorus from the Latin for gatekeeper, for its location near the outlet valve of the stomach. But the mere existence of the bacteria, or even its association with ulcers, was not proof enough that it caused gastritis. Koch's third postulate stipulated that to be classified as a bona fide causal element for a disease, an organism needed to recreate the disease when introduced into a naive host. Marshall and Warren inoculated pigs with the bacteria and performed serial endoscopies, but the pigs, 70 pounds of porcine weight that did not take kindly to weekly endoscopies, did not sprout any ulcers, and testing the theory on humans was ethically impossible. How could one justify infecting a human with a new uncharacterized species of bacteria to prove that it caused gastritis and predisposed to cancer? In July 1984, with his experiments stalled and his grant applications in jeopardy, Marshall performed the ultimate experiment. On the morning of the experiment, I omitted my breakfast. 2 hours later, Neil Noakes scraped a heavily inoculated 4-day culture plate of Heliobacter and dispersed the bacteria in alkaline peptone water, a kind of meat broth used to keep bacteria alive. I fasted until 10 a.m. when Neil handed me a 200 milliliter beaker, about 1/4 full, of the cloudy brown liquid. I drank it down in one gulp, then fasted for the rest of the day. A few stomach gurgles occurred. Was it the bacteria, or was I just hungry? Okay. Talk about taking matters into his own hands. Or maybe taking bacteria into his own stomach. Okay. Marshall was not just hungry. Within a few days of swallowing the turbid bacterial culture, he was violently ill, with nausea, vomiting, night sweats, and chills. He persuaded a colleague to perform serial biopsies to determine the pathological changes, and he was diagnosed with highly active gastritis, with a dense overlay of bacteria in his stomach and ulcerating craters beneath, precisely what Warren had found in his patients. In late July, with Warren as co-author, Marshall submitted his own case report to the Medical Journal of Australia for publication. A normal volunteer has swallowed a pure culture of the organism, he wrote. The critics had at last been silenced. Heliobacter pylori was indisputably the cause of gastric inflammation. The link between Heliobacter and gastritis raised the possibility that bacterial infection and chronic inflammation cause stomach cancer. Indeed, by the late 1980s, several epidemiological studies had linked H. pylori-induced gastritis with stomach cancer. Marshall and Warren had, meanwhile, tested antibiotic regimens, including the once forsaken alchemical agent bismuth, to create a potent multi-year drug treatment for the H. pylori infection. Randomized trials run on the western coast of Japan, where stomach and H. pylori infection are endemic, showed that the antibiotic treatment reduced gastric ulcers and gastritis. The effect of antibiotic therapy on cancer, though, was more complex. The eradication of H. pylori infection in young men and women reduced the incidence of gastric cancer. In older patients, in whom chronic gastritis had smoldered for several decades, eradication of the infection had little effect. In those elderly patients, presumably the chronic inflammation had already progressed to a point where its eradication made no difference. For cancer prevention to work, Auerbach's march, the prodrome of cancer, had to be halted early. That was our last break, but we will keep going. Although unorthodox in the extreme, Barry Marshall's experiment, swallowing a carcinogen to create a precancerous state in his own stomach, encapsulated a growing sense of impatience and frustration among cancer epidemiologists. Powerful strategies for cancer prevention arise clearly from a deep understanding of causes. The identification of a carcinogen is only the first step towards that understanding. To mount a successful strategy against cancer, one needs to know not only what the carcinogen is, but what the carcinogen does. But the set of disparate observations, from Bloomberg to Ames to Warren and Marshall, could not simply be stitched together into a coherent theory of carcinogenesis. How could DES, asbestos, radiation, hepatitis virus, and a stomach bacterium all converge on the same pathological state, although in different populations and in different organs? The list of cancer-causing agents seemed to get, as another swallower of unknown portions might have it, curiouser and curiouser. There was no precedent in other diseases for such an astonishing diversity of causes. Diabetes, a complex illness with complex manifestations, is still fundamentally a disease of abnormal insulin signaling. Coronary heart disease occurs when a clot, arising from a ruptured and inflamed atherosclerotic plaque, occludes a blood vessel of the heart. But the search for a unifying mechanistic description of cancer seemed to be sorely missing. What, beyond abnormal dysregulated cell division, was the common pathophysiological mechanism underlying cancer? To answer this question, cancer biologists would need to return to the birth of cancer, to the very first steps of a cell's journey towards malignant transformation, to carcinogenesis. Oh, I was stammering all through that. I was fucking stammering. Okay. We're at 286. But we'll stop here because my throat is fucked and I'm quite hungry. I haven't eaten breakfast. And I clearly need to work on my reading skills today.